Okay, so our goal is to have monophyletic groups. So the goal is to try and figure out who's related to whom, who are the ancestors, and how does this all work. So how do we go about actually doing that? How do we decide where the branch points should go and whatnot? Um, that's what's next. So we build what's called a cladogram. Now, the key to a cladogram is that it's only showing the pattern of relatedness. It doesn't show the specific time frames. Phylogenetic trees show how much time has passed between when different groups branched off. Cladograms just show the pattern of relatedness with no information about when things branched. So to build this, you need to collect some data about different characteristics of different groups. And cladograms are based on what are called shared derived characteristics. This is a trait found in all members of a clade, but not in the ancestor. And so here we have a list of characteristics of vertebral column, hinged jaws, walking legs, amnion, etc. Um, and so what you do is you look at the different groups and you give them either a 1 or a 0 depending on if they have that trait or not. And so a vertebral column, well, something called a lancelet doesn't have that, so you give it a 0. All these other groups, lamprey, bass, frog, turtle, leopard, has one, so you give them a 1. Hinged jaws, etc. You do that 0, 1 thing. And then you identify what's called an outgroup, which is a group that diverged before the lineage that we're studying came about. So the lancelet doesn't have any of the shared derived characteristics listed, so they are our outgroup in this particular example. And then you start to put this together. You say, okay, the vertebral column was found in the lamprey, the bass, the frog, the turtle, leopard. That must be the most ancient shared derived characteristics. That probably came about first because everyone has it. So if this is our common ancestor right here, it branched into the lancelets, which is our outgroup, and then the vertebral column must have evolved here, been present in this common ancestor, and passed on to all of these groups. The next trait is hinged jaws. The lamprey doesn't have that, but everybody else does. So the lamprey probably branched off here, and then hinged jaws evolved in this line, was present in the common ancestor, and passed on to everybody else. Four walking legs is next. It evolved here, was present in this common ancestor. And so you can slowly but surely make your cladogram. And we assume that the simplest tree is the correct one, which is called maximum parsimony. One thing to keep in mind is that cladograms are what are called dynamic hypotheses, which means that they are constantly being reevaluated. When we saw that one with the protists, I kind of talked about how based on just cell structure, anatomical, so to speak, data of cells, scientists had kind of just threw this category called protist together, and then as molecular data became available, we realized, that wasn't quite right. And so DNA sequencing is really reshaping phylogeny in many groups, not all. Larger vertebrates we were able to piece together for the most part with anatomy. So the final thing is to just kind of look at the different systems that have existed over the course of scientific history here. And it's interesting that Linnaeus, Carolus Linnaeus, who first sort of started us down this journey, he initially only had two kingdoms, plants and animals. And he kind of threw everything into one of those two groups, plants and animals. Eventually, by 1969, that we realized had some problems. For example, fungi. Linnaeus sort of called them plants, but turns out fungi are more closely related to animals than plants. They don't photosynthesize, for example. They release enzymes and break down organic matter. They eat, so to speak, like we do. So that became its own kingdom. That those single-celled eukaryotic cells became the protists, that kind of junk drawer kingdom that we weren't sure what to do with, and then bacteria, which were the prokaryotic cells, became a kingdom, Monera. Well, later, that turned into six kingdoms. 1977, my birthday, by the way, we realized that there's actually two very different groups of monorans. Some prokaryotic cells are very, very different. We called them archaea, and then another group, bacteria, and then kept protist, fungi, plant, animal. And then finally now, people realize that those two groups of bacteria are so different from one another that they deserve to be separated even further than the level of kingdom. So then in 1990, we developed the current system, which is 3 domains: bacteria, archaea, and eukaryota, which includes the plants, the animals, the fungi, and then those various protist groups. What this is saying is that you and I have more in common with a mushroom or a single-celled eukaryotic cell in pond scum than these two groups have with each other. And you might think, how can that be possible? The reason why is because these two actually have different ribosomes than one another. They have different membrane phospholipids than one another. Like the basic molecules that build the cells are fairly different between these groups. And so since they have such fundamentally different molecules, we lump them into 2 separate domains. Also, DNA evidence shows a lot of difference between these groups. So one final thing, so that kind of shows how these are dynamic hypotheses and have changed from the 2 kingdoms to the 5 to the 6 and now the 3 domains. The final thing is that, like any good hypothesis, they can be used to make predictions about other groups. And so an example of this is with dinosaurs. People have made predictions about dinosaurs because of similarities between birds and crocodiles. Here's the common ancestor to crocodiles, birds, and in the middle are these dinosaurs. And remember, birds are technically considered dinosaurs. We call them avian dinosaurs, based on this. They're monophyletic with the dinosaurs. And so if there's something in common between crocodiles and birds, chances are good these dinosaurs had it. So an example is the 4-chambered heart. Lizards and snakes and other reptiles have a 3-chambered heart. Crocodiles are kind of unique among reptiles that they have a 4-chambered heart. Birds have a 4-chambered heart. So we think dinosaurs probably had a 4-chambered heart. Another example is parental care. Birds, I'm guessing you're aware, exhibit quite extensive parental care. You may be surprised to know that crocodiles exhibit parental care. Here's like little babies. The mothers will carry them around in their mouth and move them from place to place. And so, again, based on that, we think these dinosaurs exhibited parental care. So these phylogenetic trees are important to be able to read and interpret. They can be used to make predictions about things. And like anything in science, are always subject to revision as new information becomes available. That's it. Take a look at the homework packet for this section on phylogeny, and you should be good to go.